One.
Hi, thank you for the great introduction. My name is Hayrunnisa Bolay. I'm a professor at Gazi University in the departments of Neurology and Algology. And I'd like to thank the organizing committee for inviting me. And it's a great honor and pleasure to speak in the second Dubai Neurology Congress. Today, I will talk about headache mechanisms and giving different conditions and models to understand the mechanisms of headache and head pain. Before start, I'd like to share, it's my disclosure. I have nothing to disclose financially. Okay, how do we understand headache? We need a simple conceptual framework for headache development. And we also know that headache symptom denotes a nociceptive activation of trigeminal neurons, which reside in the trigeminal ganglia and its peripheral axons innervate the cephalic structures. And among them, the outer layer of meninges, the dura mater, has the prime importance. And this system constitutes the peripheral system. And anything external or systemic triggers evoke activation of trigeminal vascular afferents, and they transmit the signals to the brainstem nuclei. The other name is trigeminal cervical complex. And at the same time, we know that there are central mechanisms, mainly in the cortex, like cortical splitting depression or thalamocortical oscillations or any disruption of this system. And descending pain control system starts from the cortex, orbitofrontal cortex, and includes hypothalamus, periaqueductal gray matter. And there is a balance between them. And these two systems, central and peripheral mechanisms, are connected. And which mechanisms link these structures? And I will today talk about at least, three examples. And one is the inflammatory substances and neuroimmune crosstalk. And in three different models, in nitroglycerin model, cortical spreading depression, and recent, pandemic, COVID-19, is probably inducing headache through these mechanisms. And also talk about the involvement of thalamus, any disruption of thalamus, cortical circuitry and disruption of the sensory processing. And also briefly, I will talk about the interaction of food triggers and analgesic abuse. So trigeminal activation is associated with neuro-vasculo-inflammatory process, and these peripheral axons in the dura mater, when they are activated, they release neuropeptides, mainly calcitonin gene-related peptide, PACAP, and substance P. And those neuropeptides have receptors on the other cells, vascular cells, smooth muscle cells, which induce, vasodilation, leaky vessels, and edema. And also some receptors are on the A-delta fibers and more, further enhance the trigeminal nociceptive impulses to the cerebral cortex. And also immune cells like mast cells and macrophages, they are all players in that system. And neuronal activity, sensory neuronal activity is coupled to the inflammatory signals, neuroimmune reciprocal crosstalk happens. And in that sense, we need to see that, any dural vessel is in close proximity with trigeminal nerve endings, with neuropeptides, macrophages. These are resident macrophages. These are not recruited from the peripheral blood. Mast cells, also they are resident in the dura mater and meninges and other nerve fibers. And pro-inflammatory mediators and cytokines, they are all identified triggers for trigeminal nociception. So they have all reciprocal crosstalk in between. And let's see the, one of the triggers, one of the provoking agents, which is consistent and reliably induce headaches in migraine patients. This is glyceryl trinitrate, in brief GTN. It's a pharmacological agent, and still commonly used to trigger migraine attacks, also in migraine and other primary headaches like cluster and PTH. It induces brief, flushing and brief headache in all subjects, but only induce migraine headaches in migraine patients, and it is around 80%. It's very high percentage, and it's a reliable trigger for human provoking studies. It is an anodonor, and it includes the guanylate cyclase and includes in the cyclic GMP increase and phosphorylation processes, and these changes activates vasodilation in the smooth muscle cells. And also we know that these GTN-induced changes affects the immune cells in the experimental models, but none of the study has showed any direct activation of trigeminal neurons. And this is one of the good examples that high percentage of nitroglycerin provoking, migraine provoking mechanism is indirectly activates the trigeminal neurons, but there is a direct activation of vascular cells and immune cells. So in the periphery, in the dura mater, GTN activates dural macrophages. Here is the IL-1B expression within half an hour and maximum at 1 hour. You can see that there is an increase in the dural tissue, these inflammatory cytokine, and also that is associated with transcriptional factor activation, NF-kappa D activation, and NF-kappa D P65 translocation to the nucleus. And these changes occurs not in other cell type, but only in the resident macrophages. And within several hours, we can also observe that one of the important cytokines, IL-6, induction in the same cells, in the same macrophages. And we also know that when the IL-6 is activated and released from these immune cells, it is important nociceptive action, and it activates dural trigeminal afferents and induce headache-like behavior. You could see the co-localization of IL-6 and these, ED2 positive macrophages. And also in later time points, there is an INOS expression, which is important for the sustained pain response. And these changes in the dura mater is also associated with mast cell degranulation. So another immune cell is also participated in the periphery. And not only in the peripheral tissues like dura mater, nitroglycerin also activates brainstem microglia. And this is the brainstem trigeminal nucleus, and you could see the IL-1 beta and NLRP3 inflammasome activation in the microglia. And release of IL-1 beta has its receptors in the neurons, and activation of neurons leads to the CFOS expression and pain perception. So it is a good example of these neuroglial crosstalk via NLRP3 inflammasome and IL-1 beta. We also know that NLRP3 activation yields other neuroinflammatory cytokines, and that leads to the amplification of tissue inflammation. So both peripheral tissues, peripheral mechanisms, and central mechanisms are activated. And recent study also showed that, also confirmed the NF-kappa D activation in the brainstem trigeminal nucleus. This is the second order pain nucleus, and increased CGRP mRNA associated with pain behavior. But novel thing is the one of the cytokines which is associated with chronic inflammatory conditions were shown in the brainstem and activated by nitroglycerin induction, and probably its access from the periphery to the central nervous system is increased by GTN. So we have, more clues, more evidences that GTN induces both peripheral and central nervous system nociceptive substances. It induces pro-inflammatory cytokines, molecules in immune cells and in microglia, which indirectly activate the trigeminal nociceptors in the dura mater and also in the trigeminal nucleus caudalis. And vascular, glial, and immune cell activation, release of inflammatory molecules that mediate neuroglial or neuroimmune crosstalks, at the end collectively induce headache development. And the second example is during the pandemic conditions lasting for the last 2 years, more than 2 years. Now, the headache is the most frequent neurological symptom of COVID-19, and it can be seen as an isolated symptom, or it can be the presenting symptom of COVID-19, which may precede fever or cough. New onset, unrelenting headache in the early course of the illness is, characteristic, and it can, and it can have features like migraine, and also it can have some features reminding the secondary headaches. But the important part is, if it is a headache as an onset symptom after the fatigue, the presence of headache at the onset is the second risk factor for long COVID. So that may help us to understand the inflammatory or neuroimmune crosstalk mechanisms, and could it be something like this? And we know that dysregulated inflammation plays an important role in COVID-19 pathogenesis. And when the SARS-CoV-2 binds to the ACE2 receptor for cell entry, that induces transcriptional responses, activates NLRP3 inflammasome, and cleave and release pro-inflammatory cytokines, and also release of damage-associated molecular patterns, and HMGB1 is one of them. In the end, they collectively induce, augment inflammation, induce inflammation, pyroptosis, and also coagulopathy. And we also noticed that the risk factors and comorbid disorders of COVID-19 are different from other respiratory viral illnesses. And these infections disproportionately impact on the patients with hypertension, like 68%, cardiovascular diseases, then compared to the allergic respiratory diseases such as asthma. And we know all these factors are also risk factor for migraine patients. But what connects these comorbidities and risk factors, the ACE2 seems to be the main mechanism connecting these comorbidities with the COVID-19. And angiotensin 2, through its receptor, induces inflammation, reactive oxygen species, nitric oxide increase in NF-kappa B transcriptional factors, vasoconstriction and nociception. And its detrimental effects are balanced with the ACE2 enzyme, which cleaves and produce, angiotensin 1,7, and through its receptors, it just counterbalance and reduce inflammation, reduce nociception, and increase vasodilation, and also reduce these reactive oxygen species and NO/NF-kappa B, and also, they have opposite effects on the NLRP3 inflammasome complex. When the virus binds to the ACE2 and this inhibits this arm, we see the further induction of NLRP3 inflammasome induction and release of cytokines as a consequence. And these cytokines in the near vicinity or in the systemic circulation could easily activate the trigeminal nerve and trigeminal vascular activation, and leads to the headache. So unbalanced angiotensin system contributes to the inflammasome activation. And in the first wave of the pandemic, we were able to identify some, risk factors and some determinants for headache phenotype. There was a robust inflammatory response, which determines the headache severity, duration, and its treatment response. The, high levels of IL-6 was associated with paracetamol unresponsiveness, and which also increased the headache days and headache attack duration and the severity. But in the milder cases, there is a moderate or mild increase of IL-6. Knowing this, we also further investigated the other inflammatory substances and inflammasome proteins, because either the virus itself or release inflammatory substances from the circulation could influence and could induce headache at the periphery, at the trigeminal vascular system. Our results showed that the HMGP1, which is a dump protein, which is both it's pro-inflammatory protein and also nociceptive protein. It's it induced nociception in the dorsal root ganglia and the trigeminal ganglion neurons. So it's significantly increased in COVID-19 patients with headache compared to the COVID-19 patients without headache. In parallel, we detected the NLRP3 inflammasome, IL-6 levels were increased, and ACE2 levels were increased. Surprisingly, we did not see any increase in the CGRP levels. Actually, there was a reduction in the CGRP levels. So the differentiating ability of HMGP1, both its sensitivity and specificity for headache in COVID-19 was significantly higher than the others, and therefore that was important. If it is combined with the others, it is really important for differentiating the headache. When we did the network analysis, the headache was positively and strongly correlated with HMGP1 levels and also NLRP3 levels and IL-6 levels, not for the other ones. And I think the HMGP1 was also negatively and strongly associated with paracetamol response. So if it is higher, there was a paracetamol unresponsiveness, and hospitalization and headache duration was also associated with NLRP3. So we have a very nice connection as a nociceptive and pro-inflammatory molecule could play a role in persistent, unrelenting headache associated with COVID-19. We also know that, cortical neurons, sensory neurons, astrocytes, endothelial cells, they all release HMGP if they are exposed to the cytokines of nerve injury. And we know during the spreading depression, HMGP1 is released and is able to induce trigeminal nociceptors. And HMGP1 mediates nociceptive action in sensory neurons through its two receptors, which also includes the NF-kappa B activation and release of cytokines. And we also know in the trigeminal system, its importance was shown in the pain behavioral studies and also neutralizing antibody studies. In conclusion, we can say that COVID-19 headache is an outstanding example of how neurovascular-inflammatory mechanisms take place in trigeminal activation and will provide better understanding of primary headaches. And COVID-19 headache is associated with stronger inflammatory response without any change in the circulating CGRP levels. And we can assume that pro-inflammatory cytokines and inflammatory proteins, including HMGP1, mainly released from respiratory tract cells, may convey to the damaged signals to the sensory neurons in the trigeminal ganglia, trigeminal perivascular nociceptors within the dura mater, and they activate trigeminal system. And therefore, I think there is a good reciprocal signaling between cytokine generating cells and trigeminal neurons. The third example is the cortical spreading depression. As we know that it's a, neuroglial hyperexcitability followed by a neuronal silence, which propagates through the cortex. And the cortex, cerebral cortex, is pain insensitive. There is no trigeminal nerve innervation, but the overlying meninges and dura mater is very, densely innervate the substances, particularly the, vasculature. And therefore, the spreading depression, its mechanism, how it's activate the overlying meninges is important. But the first mechanism is periphery, from the cortex to the periphery, how these, substances are released and activate meningeal trigeminal nociceptors. What we see, it was the cortical spreading depression in the cortex, which release, all those substances, vasoactive and nociceptive substances. And we will see it again. This is the, propagating of, the very brief one was the propagation of CSD. Now, during the 1 hour of, imaging, you could see, we only see the activation of dural vessels, meningeal, dura mater, the middle meningeal artery, and activation of, meninges, not the cortex. And this activation is totally dependent on the trigeminal nerve and associated with leak and protein extravasation in the dura mater. And also CSD was shown to induce HMGP1 release from cortical neurons that is also associated with pain response, and also induce mast cell degranulation. So those changes at the periphery, at the overlying dura mater, overlying meninges, following the CSD, is also reflected, is also transmitted to the second order pain nuclei. And this is the activation of CFOS. And you can see that during the CSD increased, CSD induce activation of these nociceptive neurons, and that can be inhibited by sumatriptan. So we have evidence that CSD can activate through the peripheral route to the peripheral trigeminal vascular system and activates the brainstem trigeminal nuclei. But we also know there are several studies showed that CSD can disinhibit subcortical diencephalic structures and activate trigeminal nucleus caudalis through the central pathway. Lambert showed that these nociceptors and the activation of TNC can occur before any peripheral activation of trigeminal vascular system. And these descending, or disinhibition could involve thalamus, and that can be, shown in both, anesthetized animals or awake behaving rodents. And so there is a top-down mechanisms also, particularly in awake animals. And in this matter, the thalamus, involvement of thalamus is very important. 6 years ago, we published that thalamic reticular nucleus is invaded, is involved by CSD. And this involvement occurred only during the, if the experiments were conducted in awake animals. It was, very susceptible to the anesthetics. So you can see the CSD waves in the cortex, and you can see what we recorded from the thalamic reticular nuclei, and they can easily propagate, and you can see the activation of cells in the thalamic reticular nucleus. Why it is important? Because thalamic reticular nucleus is a gatekeeper nucleus that controls the sensory outflow from peripheral structures to the cerebral cortex. If there is a problem like CSD in that nuclei, that cannot function, and that leads all the sensory, flow to the cortex. Enhanced sensory reaction can occur. A recent study also showed that in addition to the thalamus reticular nuclei, ventroposterior medial nuclei, which is the third order relay nuclei of trigeminal pain, is also activated by cortical spreading depression. That is also very susceptible to the anesthetics, and during the anesthesia, that did not happen, that did not occur. But during the awake behaving animal, cortical spreading depression waves are propagated to the VPM, and there is a propagating activity within the limits of cortical spreading depression. So in conclusion, CSD can activate trigeminal system both peripherally and centrally, and inflammation and activation of neon cells are involved in this process, and also involvement of thalamus, particularly the thalamus reticular nuclei and VPM. These are two critical nuclei, not only for headache, but also other sensory associates of migraine and its relations with sleep. And in that regard, I would like to give another example how the food triggers intersect with analgesics. We identified that sulfotransferase enzyme inhibition is important. These enzymes detoxify drugs and chemicals, and they play a role in the metabolism of neurotransmitters. And we know one of the most important food trigger is citrus, and the hesperidin is one of its ingredients. And the non-steroids, they also they are both substrates and also inhibitors of these enzymes. And using this model, we were able to show that they both decrease the CST thresholds and mechanical thresholds, yielding pain behavior. And at the same time, SULT1 activity, enzyme activity, was significantly lower. So now we have another, player in both periphery and in the brain tissues. SULT1 inhibition could be a common mechanism which food triggers and analgesic abuse induce chronic daily headache. And I would like to talk about lastly with the disruption of sensorimotor processing, because in the sensorimotor integration, we were able to show that there is a reduced inhibitory circuitry in the sensorimotor cortex, which was shown by Saib paradigm, and there is a somatosensorial temporal discrimination problem, very prolonged sensorial discrimination. And here is the example of SAE. It is a paradigm if a preceding peripheral stimulus artifact, induction of median nerve, the TMS-induced MEP is inhibited by the peripheral stimulation. This is the normal reaction we observed in the healthy volunteers and also in the migraine without aura patients interictally, but during the ictal phase, this is inhibited. The inhibition is reduced, and psi is, psi is reduced during the attacks. And reduced inhibition and increased excitability of the sensorimotor cortex is much more prominent in the pre-ictal phase compared to the ictal phase. And actually, we have 3 longitudinal studies in cases. You can see that the much more significant excitability occurs, or disinhibition occurs, several hours before the onset of headache. So that means something is going on in the somatosensorial cortex or thalamocortical network before patient feels any headache. In the migraine without aura patients, these are not migraine with aura patients. And also, we were observed that the consecutive somesthetic stimuli, differentiating them is also prolonged, is disrupted, and significantly prolonged during the attack in the episodic migraine patients, or stayed, sustained this prolongation in the chronic migraine patients. So, what caused that disruption? what disrupt the sensory gating then in migraine patients? We know that migraine headache is not a simple transfer of impulse from the peripheral trigeminal nociceptive induction, and unless it is allowed from a top-down mechanism, none of the peripheral stimuli can reach to the cerebral cortex. So one theory is the cortical spreading depression, which is totally different in human brain. It is limited and it cannot, propagate to the long distances, but it is able to, spreading depression in human cortex is able to interrupt the thalamocortical network or any talk between the, first order, higher order oscillations in the thalamus and cortex. And that will be important in inducing, sensory discrimination and impairing the sensorial integration. And we know there is a multisensorial disruption in migraine, both visual, auditory, tactile stimuli, as well as the headache. They occur at the same time. And in the one of the clinical studies, it was shown that during the aura, there is a coexisting headache and photophobia, phonophobia, and nausea. So the involvement of reticular thalamic nuclei or any other higher order thalamic nuclei could be important in that perspective. And, we could say higher order thalamic nuclei, such as this pink here, can function as a hub, which synchronize distant cortical, cerebral cortical areas via, transthalamic pathway, transthalamic connections. And CST in one of the circuit can cause neuronal dysfunction in the remote areas within the same hub. And such a dysfunction in one hemisphere would definitely impair the other hemisphere and disrupt large-scale functional brain networks that we detect, that we observe in the fMRI studies or resting MRI connectivity studies. So as a conclusion, trigeminal nerve fiber innervation of cephalic vessels and their close proximity to macrophages, mast cells, and glial cells, they constitute the neurobiological basis of neurovasculo-inflammatory process during headache, and that can be induced by intrinsic and or extrinsic factors, including migraine triggers or SARS-CoV virus infection. And we also know ST in the cortex can activate peripheral trigeminal nociceptors, or it can propagate to the thalamus and disrupts thalamocortical network. It interrupts sensory gating and results in headache and other sensory problems. I would like to thank my collaborators and my team. I couldn't put all of their names, and I would like to thank you for your kind attention.